Circadian rhythm. It's the natural process that helps control your daily schedule for sleep and wakefulness. It regulates the sleep wake cycle, repeating itself on each rotation of the Earth, roughly every 24 hours.
Our lab is very interested in the circadian clock. We happen to be looking at genes in the brain that are controlled by the circadian clock. And we found this particular gene called Chi301, which we didn't know what it was or what it did at the time, that seemed to be strongly controlled by the circadian clock in the brain.
Dr. Eric Music's lab at Washington University School of Medicine in St. Louis made a discovery explaining a possible link between circadian rhythm dysfunction and Alzheimer's disease.
It just so happened the gene that encodes a certain protein called YKL40, which our colleagues right down the hall in the Alzheimer's Center had been working on for many years as a biomarker of Alzheimer's disease.
With Alzheimer's disease, fractured sleep, daytime sleepiness, and other signs of disturbance in circadian rhythm are common complaints.
A lot of patients that have Alzheimer's, their sleep-wake cycle breaks down. They tend to sleep more during the day, to be awake more at night. Some of them are confused about day versus night. And there's plenty of data showing that the circadian clock, for some reason, is not working properly in Alzheimer's. The circadian clock controls when we sleep. And if you don't sleep enough, you can accumulate this protein called amyloid beta in the brain that forms these amyloid plaques, which we think are kind of the first hallmark of Alzheimer's disease.
It only gets worse as the disease progresses. The associate professor of neurology says the protein called YKL40 offers a clue as to why this happens.
To investigate the role of what YKL40 is possibly doing in the disease, we took a mouse model of beta amyloidosis. So what that means is they get amyloid beta plaques in a similar way that humans get amyloid beta plaques. It's not exactly an Alzheimer's model, but it recreates one aspect of Alzheimer's disease, and which we think is one of the primary drivers of the disease. And so we took these mice and we deleted Chi301 gene, which means they don't create any YKL40 protein. And so when we take away YKL40, we found that those mice develop plaques at a slower rate, which suggests that YKL40 is perhaps playing a detrimental role in the progression of amyloid beta plaques.
Alzheimer's disease is characterized by chronic inflammation, so they wanted to closely observe what's happening in the brains of genetically modified mice lacking YKL40. They discovered effects on immune cells in the brain called microglia.
The basic idea is that the microglia are supposed to be playing a protective role. Sometimes microglia can play a beneficial role in Alzheimer's disease by eating away the amyloid beta plaques, but they can also sometimes produce more inflammatory factors.
We think that YKL40 serves as a brake to kind of keep microglial activation and inflammation under control. But in doing so, it may suppress the ability of these cells to eat up this amyloid beta and these plaques. And so we think if you have too much YKL40 around, it may kind of prevent the microglia from properly taking up A beta and removing these plaques.
What's unique about our study is that we're the first ones to show that it actually does affect microglia in terms of how they uptake amyloid beta, helping to establish the relevance of YKL40 in how the disease progresses.
A related study at Washington University about a genetic variant in people with Alzheimer's complements Music's research.
The people that had this particular genetic, it's called a polymorphism. So about 20% of the people have this specific genetic mutation that makes them have less of the YKL40. And so those people that genetically have less seem to have a slower progression of their Alzheimer's disease. So they got worse at a slower rate. Much like in our mice, when we deleted this gene for YKL40, the mice had less plaque. It suggests not just a marker of the disease, but may actually play a role.
Which offers hope that therapies may target the YKL40 producing gene, which is linked to the circadian clock. This may slow the course of Alzheimer's disease, but a lot of work would need to be done to make sure this approach is both safe and effective.
So in theory, one hypothesis, we don't know this for sure, but it's possible that if you had a drug that lowered levels of YKL40 in the brain, it could kind of promote microglia and their ability to eat up these amyloid plaques and maybe reduce the amount of plaque. But that still has to be proven. I think we envision it as something that if you could treat people before they have symptoms and help to eliminate these plaques from their brain, that they could, maybe never develop the disease or develop a much milder case later in life.